I'm Giancarlo, a master's student supervised by Elvira Saraiva, and we are doing the characterization of monocytes DNA extracellular traps that's induced by SARS-CoV-2. In this work, we aim to verify whether SARS-CoV-2 and its spike protein induce the release of DETs in human monocytes, characterize the morphology of DETs, study the stimuli mechanism involved in the DETs release, investigate the cytotoxicity of DETs to lung epithelial cells and to endothelial cell line, analyze the thrombogenic role of DETs induced by SARS-CoV-2. Here we show that SARS-CoV-2 and its spike protein induce the release of DETs in human monocytes. In this picture, we show the morphology of DETs, visualizing the DNA structure stained by the API and the trapped virus stained in green pointed by arrows. And by scanning electron microscopy, we show the filamentous structure of DETs with bed-like structure that likely represent the various proteins associated to this structure. Here we show that Royce is involved in the release of DETs, which is inhibited by protein monocytes with NAC and DPI. NPO, PAD4, calcium, elastase, and gasdermin D are also involved in the DET release. DETs were toxic to calum-3 and 8,549 lung epithelial and to HBM8C endothelial cells, as shown by LDA release. Since NETs were thrombogenic, which tested the procoagulant action of DETs, our results show the DETs present greater clearing capacity compared with NETs from the same donor and induced by SARS-CoV-2. Analysis of gene expression of tissue factor in monocytes were also higher than in neutrophils. We concluded that SARS-CoV-2 induced the release of DETs in human monocytes and may be toxic to lung epithelium and visual endothelium. DETs have a procoagulant effect and can trigger thrombotic phenomena in COVID-19.